welcome back. Exciting to have our next speaker, Chioma Oselu. She will be talking about Python in Africa's DNA, building inclusive and reproducible genomic tools. The stage is yours. A huge round of applause for Chioma.
Hi everyone, and thank you so much for being here. My name is Chioma Oselu, and I am a computational biologist focused on building better tools for genomic prediction. And this is especially for African populations, which are often overlooked. The title of my talk for today is Python in Africa's DNA, Building Inclusive and Reproducible Genomic Tools. Now, a lot of conversations around inclusion in genomics usually focus on data. So, talking about who is represented in data sets, who is missing, and, what that means for discovery. But today's talk is about inclusion, and that's inclusion in design. Specifically, we're going to be talking about how writing code, regardless of how clumsy or inconsistent it is, can make you can become a way to ask better questions about fairness, reproducibility, and whose data gets to count. I'll also walk you through how I use Python to shape more reproducible population-aware pipelines for polygenic risk scores, PRS for short. So I'm going to be talking specifically in contexts where most tools silently underperform, right? So let's get into it. Now, this slide shows two snapshots. But before I get into the snapshots, let me give you some context. In genetics, we use models like polygenic risk scores, PRS for short, to estimate how likely someone is to develop a disease based on their DNA, right? So now these scores are increasingly used in research, clinical settings, and even in wellness apps, right? But there's a serious issue. Most methods don't perform well in African genomes. And now it's not because African, data is bad. Is usually because many were trained, tested, optimized on European data sets only. Now, the image to the left shows a snapshot from Nature Medicine by Fatima et al. Now, this shows the ancestry breakdown of different studies included in GWAS catalog globally. And we can see how Europeans are largely represented with populations like African, Latinos, Greater Middle Eastern having little to no data. So African populations are still significantly underrepresented despite having the highest genetic diversity in the world. The image to the right shows how African genomes, perform how the performance drops dramatically during in a PRS study. So this means that the predictions are less accurate, and if they are less accurate, that means they are less reliable and they are less actionable for African populations. Now the consequences are they're serious because they include missed signals, they include incorrect classifications, or worse, being excluded from precision health initiatives altogether. So why is that? uh, we already spoke about this issue of data. Most models are trained on European data sets, so it's quite obvious that they would only capture patterns that are specific to that population, which would include effect sizes, and that don't necessarily transfer well to other ancestries, right? So this is what I like to call data deserts in global genomics. This map shows, different regions, specifically highlighting Africa, Latin America, and South Asia, where genomic data is severely lacking. Now, these gaps is what I'm not just trying to, talk about missing data. They represent entire populations that are being left out of how tools are designed, how tools are tested and deployed. So, one solution, which are, which are currently critically looked at is H3 Africa, the Nigerian 100 Genomes project, and there are other regional efforts that are expanding the data landscape. But one thing I would like to point out is that representation alone is not enough. There are actually other factors which we'll be looking at in the next slide. Now, contributing factors include genetic factors like allele frequencies and linkage disequilibrium structures. So basically, just to, in a nutshell, just to explain what I mean by all of that, you know, genetic stock. So, for example, a variant that is common in European populations might be rare in African populations, and then these signals are. They make the models behave differently, and therefore they must be accounted for. If they're not, it can lead to you know lot of reduction in accuracies, in the accuracy of the models downstream. So there's also more. There's something we call gene environment interactions, and this involve age, sex, diet. So all of these influence polygenic risk score performance. And one solution, as previously mentioned, is better representation in dataset. But there's another, and that is actually my focus in today's talk, which is methodological redesign. How can we build tools that perform better across populations, starting from the code? So now, most pipelines are they're not they're not broken. I won't say that they are broken. Most of them were not just built to accommodate the uniqueness, right? This uniqueness that we've stated out. A few examples include assuming a variant is common because it is in one population's data, and this slide shows a workflow from the input to QC, stands for quality control, and down to the specific genomic tool, depending on the context. So, for example, the user is expecting file names or headers to follow a certain patterns. That a certain pattern, that can lead to bias and inconsistencies downstream. So applying the same quality control thresholds, even when the genetic architecture is different, can also lead to you know, uh, inaccuracies. So these assumptions, they seem minor, but they actually shape results in subtle ways. And the tricky part is that these tools, they won't crash. They will still run, but it's just that they will give weaker results. And unless you're looking closely, you won't notice the exclusion. So let's get down to my Python journey and learning. At first, I didn't start out trying to build a pipeline. I actually started with small problems, but I did know Python, or at least I was learning it, and that became my entry point. Writing small scripts, to solve small problems, using Python to clean data sets, rename columns, detect inconsistencies, and even move files from one folder to another, right? But so as I learned more, I realized that I could also use Python to control, to solve and to control bigger processes, like, for example, how tools interact. And that brings me to the building blocks. So over time, I started seeing patterns. I kept doing, I was doing the same tasks over and over again, repeatedly. So I started using libraries that gave me structure. Libraries such as argparse, pandas, subprocess, and shutil. These are the libraries I'm going to be focusing in today's talk. So I moved from running someone else's pipeline. I said to shape one of my own. Looking at argparse, argparse is used to create for my specific project, is used to create flexible command line scripts. So it made my script flexible and user friendly. Pandas, on the other hand, was used to clean and inspect massive tables, using this specific pandas library. And in genomics, we work with very large data sets. So pandas is a very useful library when working with such data sets because it helps to clean, explore, and transform the genetic data. Then there's subprocess. Subprocess happens to be one of my favorite libraries because with subprocess, I was able to use it, or I am using it to trigger containerized genomic tools. So, for example, running Docker containers plus shell commands, in Python. So there's also shutil. Shutil, I frequently use it to automate file handling, moving, organizing files during reformatting or preprocessing. As I mentioned earlier, when I started, I wasn't thinking about pipelines or systems, but I kept following the problems. So eventually, the scatter script started to, form something structured. So all of this is actually still a work in progress, and that's actually the point. I started from learning. This slide shows, just a should I call it a walkthrough or a representation of the process during the whole. Obviously it's still a work in progress, learning, frustrations, finally arranging the scripts, reusing, and then down to the reproducible genomic pipeline. So all of this is still a work in progress, and that's actually the point of this talk. That is the heart of this talk. You don't have to wait until you're an expert to build something meaningful. Start where you are with the data you have, with the part that bothers you the most. That's how we like, I think, or I believe, we move forward, and how we build tools that include more people. So this slide shows a simple visual comparing an early script I wrote to the more modular version I use now. So it's not complexity. Complexity is not the whole point. The difference is in intention. Using code to solve problems and making your pipelines reproducible, so that when you run it on your system and you push to GitHub, someone else can actually pull your codes and run it, and it produces the same result. That's the whole concept of reproducibility in this context. So this is how, to me, this is how learning starts to make sense. And representation starts in the code. I feel it starts in the code. I believe it starts in the code. So let's as developers, let's write better stories with our codes. Thank you so much for being here and for listening to my presentation. If anything I shared today resonated with you, if you're building tools, if you're thinking about inclusion, or just, finding your path in tech, or specifically computational biology, I'd love to stay connected. You can find me here on LinkedIn. You can do that by scanning the barcode, and it will take you straight to my LinkedIn page. And to the right, I have a list of few public resources that I've found helpful in my journey, along with some of my articles. Thank you again to the PyLadies Con team for creating spaces like this. I'm super grateful to be a part of it. Thank you.
That was an incredible informative session. Thank you so much, Sharma. And your highlights on what data deserts in the global genomics look like is kind of an eye-opening for me as well. Thinking how various tools that we build and design could possibly lack um, the right set of understanding just due to that it's not diverse enough. That's the tool is built upon itself. Thank you so much again. And stay tuned, our lovely audience. We have incredible, exciting sessions coming up. Thank you.